Tēnā koutou, tēnā koutou katoa. Welcome here to Braintree, to our MS in Parkinson's Canterbury presentation today. We're running this Keynote series as a way of communicating some of the newer kind of research and other things that are of real interest to our community. So we're really grateful to host Associate Professor Tracy Melzer here today. I'm going to tell you a little bit more about him shortly, but before I do, I just have to do some of the health and safety type stuff that we have when we have a group of people. So we have the emergency exits here in the building are at the front where you probably came in, and there's one just out by the, past the cafe, out onto the deck. There's also one at the back there, just on the side. If they're in the event of us needing to evacuate at all, the meeting point is out very unsalubriously by the rubbish bins or the bike stand, depending on what you see first. In that area, and yeah, I'm sure none of that's going to need to happen, but it's something we need to tell everybody about. I'm Linda Dockrell. I'm one of the health and wellbeing advisors that we have here at MS and Parkinson's. There is three of us all together, along with the rest of our amazing staff who helped us get this event together today. So I will introduce you to the person you've come to see today. And this is Dr. Tracy Melzer. He's a research professor in the Department of Medicine at the University of Otago and the imaging research manager at the New Zealand Brain Research Institute, which many of our community have got connections to or often participate in the research that goes on there. Dr. Melzer is interested in the application of brain imaging techniques, so things like MRI and PET and all those alphabet soup things, and it helps to understand health and disease, so using those techniques. His interests are broad, but his primary area research focuses on cognitive impairments and the development of dementia in people with Parkinson's. I'm sure there's a bit more that you'll tell us about yourself as we go, but I'll just introduce, let Tracy take it away. Thank you.
Kia ora koutou, ko Tracy Melzer tōku ingoa, and it's my great pleasure to be here today. So thank you very much for the introduction. And as I said, it's my pleasure to tell you a little bit about what we're doing at the University of Otago, Christchurch, and the New Zealand Brain Research Institute. Now, I won't be speaking for the whole time because I brought along a special guest, Dr. Nicky Slater, who works with us at the NZBRI, and she will be speaking to you a little bit later. But I will start things off. And as you heard, I like to think about taking pictures of people's brains and trying to extract information from those pictures and hoping that it can tell us more about disease, about health, about aging, and trying to use those to direct us into new therapies or essentially assessing how those new therapies are working. Now, one of the techniques that we use is Positron Emission Tomography, PET imaging, which is what I'd like to focus on today. So, as I said, I do like brain images, and I tend to look at brain images a lot, and I love spending my time looking at these beautiful images. Now, the two main types of imaging that I spend my time thinking about, as I mentioned, are MRI, magnetic resonance imaging, which you can see an image of over there on the left, and as I just mentioned, positron emission tomography, or PET imaging, which is over on the right. Now, on your way in, you said that you were going to be picked on with difficult questions. So I'll open this to the entire floor. Does anyone have any idea what the main differences are between those two different techniques? Between magnetic resonance imaging and positron emission tomography? Any guesses? Okay, here's your question, or here's a hint. One of those techniques uses powerful magnets. Which one do you think? Exactly, MRI. That's right. Now the other uses a radioactive material to help create the image. That's not MRI, that's positron emission tomography. So a very clever use of radiation to allow us to look inside the body. Now, both of these techniques are fabulous, and they can both give us useful information about the brain, particularly in our case the brain, but also the body. And so I'd like to focus in on positron emission tomography. So, I will ask for a little bit more participation here because we have a question. We have a potential to go one way in this talk, or we can go another way in this talk. So here's the question. Would you like a quick overview of how PET imaging works?
Yes, please.
Oh, good. I was hoping you would say that. Right. So PET imaging is incredible, and as far as I know, it's the only application of antimatter that I can think of. Okay? I'll get to why I say that. So, this is a relatively complicated slide, but we'll run through it together. So, the first step in PET imaging is that we use a cyclotron or a generator. We need to create radioactive material, and it has to be a specific type of radioactive material. So that's what we see in step one. We need to produce this radioactive material. Second step, which is where the very clever chemists come along, is that we need to find some, what you see here, they call this a precursor. You need to find some chemical which will bind in the body to whatever we're interested in. So for example, I will use the protein called amyloid, which we'll get to in just a second. But there's amyloid, let's say, that we want to image in the brain. The first thing that we need to do is get our chemists to find some chemical which will bind to the amyloid, okay? So that's what we call creating this precursor, this finding this chemical which does exactly what we need to do. The next step is that we then take our radiation and we stick it on to that chemical, right? So now we have this chemical which goes exactly where we want it to go, but now it has a passenger, and that passenger is radioactive material, okay? So now we have our radioligand, which is what we call it. That goes through strict quality control, which always happens at various points during this process. But once we have this tracer, we can then inject it into our participant. Okay, so we'll inject this into the participant, which you see lying here on the bed, which will then enter into the PET scanner. Now, this radioligand, this chemical with a bit of radiation on it, will then flow through our blood flow, right? Flow through our circulatory system. Okay, and as it flows through, it will come in contact with whatever chemical we're interested in, amyloid, for example, and it will stick to amyloid, okay? So we see our person here, and we can have our radioligand sticking to amyloid, and then what happens is that we have radioactive decay, right? And this decays by a very specific pathway called, it shoots out a positron, okay? And what that positron is what we call the antimatter pair to an electron. So, does anyone know the definition of antimatter? The definition of antimatter is that it's the exact same as matter, except it has the opposite charge. So, we're all familiar with an electron, yes? That negatively charged particle which is rotating around our nucleus and all of our atoms. The positron is exactly the same, except it's positively charged. So what happens is it decays and it shoots off a positron. That positron will travel some distance, it will run into an electron. And when antimatter runs into matter, we have an annihilation, right? So electron comes and runs into a positron, and what happens is they turn from matter into energy, and they send off photons in equal and opposite directions. So we send out these gamma rays, essentially, in equal and opposite directions. And what we then do is we have this circular detector, our PET detector, and what we can do is we can detect when these two photons arrive at the same point on opposite sides of the detector. Then we use some fancy maths and work back to find where that exactly came from in the brain, and we can create these beautiful images of different functions in the brain. So I think it's fascinating. It uses antimatter, and it's very flexible if you get your chemistry right. And I'll come back to that in a second. So what I mean by that is, if you get your chemistry right, if you create this chemical or this molecule which sticks to whatever you're interested in, you can look at lots of different things with PET imaging. And hopefully I'll give you a few examples of of how we're using that here in Christchurch. Okay, so there's your whirlwind summary of how PET imaging works. So now the question is, why are we actually interested in PET imaging? And there's lots and lots of different answers. Anybody have a reason why we would be interested in PET imaging? What can we look at with PET imaging, for example? I'm about to tell you one of the things that I'm interested in. What was that, sorry?
Nerve cells.
Yeah, we can look at specific aspects of nerves. Absolutely, yeah. We can look at things like metabolism, so we can look at function in the brain. But the real renaissance as over the past 10 years has been the ability to look at proteins in the brain and the deposition of proteins or the accumulation of proteins in the brain. And the reason this is so important is because we now know that many of these neurodegenerative diseases that we're interested in, like Alzheimer's disease or Parkinson's disease, are associated with the deposition of proteins which become misfolded, and they'll clump together and they'll be deposited in brain tissue. So, the really exciting thing about PET imaging is that over the past 10 years, we've now developed techniques where we can use PET imaging to look at these different proteins in vivo, so in people who are still alive, right? So what I mean by that is, we have these three proteins which you see up here. We have a protein called alpha-synuclein, a protein called beta-amyloid, and a protein called tau. Now these three proteins exist in all of us, and will just be doing their things. We know some things about some of these proteins and not so much about the other proteins. But what we do know is that, for example, in Parkinson's disease, we know that alpha-synuclein will begin to misfold and misbehave and will be deposited in brain tissue. We know that in Alzheimer's disease, beta-amyloid and tau will both again start to misfold. They'll become really sticky, they clump together, and they'll be deposited in the brains of individuals with Alzheimer's disease. Now previously, previously we couldn't look at these in vivo. We had no way to measure these in vivo, but we can now look at that with PET imaging. So this is an image from late last year of the deposition of this protein, this Parkinson's protein, alpha-synuclein. The first in human example from late 2023, showing what the distribution of this protein is in an individual with Parkinson's disease. Here you have an example of amyloid PET. So this is looking at that hallmark Alzheimer protein, which we see deposited with Alzheimer's disease, and you can see that with PET imaging. And now even, more recently for us here in Christchurch, we can look at another Alzheimer protein called tau. Okay, tau is another hallmark protein of Alzheimer's disease. So, I'm speaking here at the MS and Parkinson's Canterbury Association, or Society. And the question is, why am I spending so much time talking about Alzheimer proteins? Why am I talking about amyloid protein or tau protein? So if you go back to the histology and to the at autopsy in individuals, if you look at Parkinson's patients who have developed dementia, it's suggested that up to half of those individuals will also show enough amyloid and tau to receive a secondary diagnosis of Alzheimer's disease. That's up for debate, but some have reported that in the literature. So the question that we were really interested in was to say, right, well actually, does this affect people in life? Can we actually pick up these Alzheimer proteins in Parkinson's disease here in Christchurch? And so that's what we set out to do with these new PET techniques. Can we see amyloid and can we see tau with PET imaging in individuals with Parkinson's disease? So those are the two questions that we set out to. Now obviously, we would love to be able to comment on alpha-synuclein, right, which is the hallmark of Parkinson's disease. But as I said, there was a first in human, just one example late last year, so that is still under development, and that's really the cutting edge. Hopefully within the next year or two or five, that will become available around the world. I'll definitely keep you updated as to what happens there. So unfortunately, we don't have alpha-synuclein here in Christchurch, but we do have access to amyloid PET. So this is again the ability to look at misfolded amyloid protein, which we associate with Alzheimer's disease. Now on the left, you can see what we call an amyloid positive scan, and on the right, you can see what we call an amyloid negative scan. So on the left, what you can see is bright signal. You're looking at a horizontal slice of the brain. This is as if you were to cut the brain this way and look down on the brain from above. So here's the front of the brain, here's the back of the brain, left and right. And what you can see is essentially similar bright signal throughout the entire brain. And that's indicative of high levels of amyloid in the brain. Whereas on this individual, what you can see is much less signal, much less uptake in the edges of the brain. And that's very typical of what we call an amyloid negative scan, indicating low or no levels of amyloid deposition in the brain. So slightly easier is if I color code these images. So hopefully we can all tell the difference between an amyloid. Would this be positive or negative, do you think?
Negative.
Negative, that's correct. And the one on the bottom is going to be positive. Yeah.
Are these the clumped up amyloids or?
Yes. So in the red, what you're looking at are the clumped up amyloids, those plaques that are being deposited. And what you can see is the red is indicating that those are spread throughout the brain tissue, whereas in the amyloid negative, you're not seeing that throughout the majority of the brain. So the question that we had was, how does this seem to relate to Parkinson's disease? So we scanned 115 people with Parkinson's disease, and you can see those results here. There's only 2 plots in the talk, and this is the first one, but I'll walk you through it. So on the y-axis, you're looking at the amount of amyloid. So 0 is a young, healthy individual. 100 is what you would expect to see in a typical Alzheimer patient. The filled circles are what we call amyloid positive. The open circles are amyloid negative. And this is spread across individuals with Parkinson's disease with normal cognition, those showing some difficulties with thinking and memory, and those individuals diagnosed as having dementia. Now, what you can see is that there are, most of the individuals don't show amyloid, right? Most of our individuals are amyloid negative. But there are a few who are showing levels of amyloid that you would expect to see in Alzheimer's disease. What you see here in red is the level that you would expect to see in Alzheimer's disease. So you can see that there are very few individuals showing the Alzheimer-like levels of amyloid. Now, this is still, we have an interesting question here. Our question is, well, interestingly, do we think that these individuals who are showing amyloid, could they potentially be at risk for future dementia? We don't know the answer to that, but that is what we are currently looking at, to see if there's increased risk, even if we don't see that much amyloid. If you do have amyloid, is it increasing your risk of future dementia? As I said, we don't know the answer to that, but that's what we're currently looking at. Actually, we don't really need to do this. Is just to drive home that we don't really see this amyloid in Parkinson's. So these are different types of Alzheimer's disease, and you can see lots more amyloid and much higher levels in Alzheimer's disease relative to Parkinson's. So it doesn't really look like amyloid is a driving factor in Parkinson's disease. So I now move on to tau PET here in Christchurch. Now this is a newer technique than amyloid. Now tau, remember, is another Alzheimer protein, which again misfolds, clumps together, and is deposited in the brain. And again, we were interested to know, or still interested to know, does this affect Parkinson's disease in a similar way that it affects Alzheimer's disease? So I have a story to tell you about tau PET. Tau PET, we acquired funding for tau PET in 2015, and we began our odyssey of attempting to acquire tau PET here in Christchurch. Now, this is a new ligand, so it needs to be produced in Melbourne, right? So the process for that is it's produced in a machine called a cyclotron, which creates the radioactive material. Then you have your chemistry to bind it to tau, right? To something that will stick to tau. That's done in Melbourne. Now, it's radioactive, and we have something called a half-life. What that means is that, well, with this particular radiation, the half-life is 2 hours. Okay, so that means that if we start with 100% of our tau, after 2 hours we have 50%. After 4 hours we have 25%. After 6 hours we have half of 25, right? So it's constantly decaying away. So it's always a race against time. So we need to produce this as quickly as possible, get it to Christchurch, get it into people, image those people as quickly as possible to make sure that there's actually some signal there to measure, okay? So it's always a race against the clock, and the race starts very early in the morning in Melbourne, where they produce the radioactive material in the cyclotron. They then package it up for us, shield it, put it in a little package, give it to the couriers in Australia. Those couriers in Australia make their way to the Melbourne airport. We then have to clear customs and lots of paperwork because we're putting radiation onto a plane. We then get that onto a plane. That plane then flies to Christchurch, lands without delays, hopefully. Then we give it to the New Zealand couriers. Those New Zealand couriers pick up the radiation. They drive it as quickly as possible to Southern Cross Hospital. We then grab the radiation, do a lot of quality control to make sure that it's still valid and viable. We then get that into syringes. We then get that into our participants who have come on time. Thank you very much. And we then inject. We have to wait for it to bind to tau, and then we get them into the scanner and get a successful scan. This has been a challenging process. And I can say that our success rate is unfortunately under 50% for the number of planned days for scanning and the number of successful days. Some reasons that we have not been able to do this, well, I guess.
Wouldn't it be easier to send your patients to Melbourne?
That's an interesting question. We have asked ourselves this question before. I mean, that is a lot to ask people to do. Is to get them to go to Melbourne. Well, maybe it's a great excuse to go to Melbourne.
Yeah.
Yeah, you know. Yes, we have had that conversation before. Yeah. For some people, that is a very valid option. For some people, that's not. So we have had that discussion. Yeah. Some of the reasons why this has failed is quality control has failed, the production has failed. We've had a perfectly packaged tau sit on the tarmac in Melbourne and just get left there and forgotten about. We have had a pilot refuse to take the radiation because there was a dog on the flight. We have had, again, to be honest, if it arrives in Christchurch, we're 100%. So if we get the tracer, we know what we're doing. So it's been an odyssey, but I can now tell you that we have success, and we have successfully scanned 17 wonderful volunteers. Many of these volunteers have been canceled 5 times and keep coming back. So if any of you are in the audience, thank you very much. We greatly appreciate, and we realize that it can be quite annoying. So our main question is, right, so if we have these 2 Alzheimer proteins, amyloid, doesn't look like it's making that much of a difference in Parkinson's disease, although we're still going to follow individuals over time to see if it provides a risk of future dementia. Now we want to ask that same question with tau. And what I can report to you is that we have now scanned 17 individuals with tau here in Christchurch, and we have found 1 individual who may, we're not entirely sure, but may show higher, not Alzheimer levels, but slightly higher levels of tau deposition. So on the right here, you have what we would consider to be a normal tau scan. So this is just a very low level throughout the brain, and that's what we would expect to see with healthy aging. Over here on the right, you can see two little areas of red indicating slightly higher levels of tau. The significance of this is unknown to us at this point. That's essentially why we're performing this study, to see if we're able to comment on whether this has any clinical significance at all. So that's where we currently are with amyloid PET and tau PET and its relation to Parkinson's disease here in Christchurch. And before I introduce our special guest, I might do a whirlwind introduction to something that I think is actually quite exciting. So, it's another PET technique. Another PET technique we can use is to look at metabolism in the brain. Right? So we can look at function of the brain and how well the brain is using glucose, or sugars, and see how well it's functioning. Now, this is the gold standard for measuring function in the brain, is glucose PET. But we're wondering, can we use a trick from the acquisition of the amyloid PET to get something that looks very similar to the metabolism PET? So here's what I mean. On the left, you have an example of metabolic PET. This is the gold standard for measuring function in the brain. Some of the downsides are, again, you have to have a new injection of radiation. That new injection is more time for our participants. It's more radiation for our participants. It's cost to the system. So if we were able to eliminate the need for that particular scan, but still get the same amount of information, that would be a win all around, right? So we think that we can do that with amyloid PET. And this is what I mean. So normally what happens with amyloid PET is you will inject the amyloid tracer and you will wait 90 minutes for it to stick to the amyloid, and then you'll have a 20-minute scan, where we measure amyloid deposition. But we think that if we take a picture for the first 5 minutes after we inject, so if we inject the tracer and we open up the camera and take a picture over the first 5 minutes, there's no time for that chemical to bind to amyloid. So what you're seeing is blood flow or perfusion, or what we think is a measure of metabolism. So the idea for us is, can we, immediately after the injection, take a 5-minute image, which we think is reflective of metabolism, then we wait the normal time, and we take our late phase, is what we call it, a 20-minute scan, and we get information about amyloid. So we think that with a single injection, we can get information about amyloid, and we can get information about metabolism. So that would be a 2-for-1, and it really would give us free information, right? Without extra radiation, extra cost, extra hassle for individuals. Now, this is what we think, right? And this is what we're testing. And this is what a PhD student named Will A. Is working on at the moment. So, the question is, if we use this early phase trick and we compare it to metabolism PET, functional PET, can we see a difference? So I'll leave you with a quiz. And the quiz is, one of these rows is early phase amyloid. One of these rows is the gold standard metabolic PET. This is from the same individual, and I challenge you to find a difference between these two images. Okay, I will give you the answer. There's a little bit of a difference here, and there's a little bit of a difference here, but they're pretty close. And so what we're seeing is that the association between these two images is very good, and now we're in the process of confirming to ourselves and the rest of the world that this is in fact a free information and could be very useful for not just amyloid tracers, but any tracer. And we think that it probably is. So we're working on that as well, and that could be helpful, useful additional information.
Excuse me. Why would we be interested as a Parkinson patient on the metabolic,
Yep.
glucose metabolic image?
Yep, that's a good question. So the question is, why would we be interested in metabolic information in Parkinson's disease? So this metabolic PET has been used for additional information for differential diagnosis. So if there's a challenging case of Parkinson's, if it's unknown whether that may be multiple system atrophy or supranuclear palsy, or maybe even something like frontal temporal dementia or another one of these neurodegenerative diseases, those can be associated with specific patterns of metabolic difference. And so sometimes if we have that information, it may give us more information, help with the diagnosis, put you on the right track for different treatments, different trials, that kind of information.
So for elimination.
Yep, yep. And so it could actually, this would actually apply beyond Parkinson's, right? This would apply to Alzheimer's, for example, if they're having an amyloid PET scan, particularly if that scan comes back as amyloid negative, right? If it comes back that there's not amyloid, then this metabolic information may give us more differential information about what potentially could be causing any symptoms that we see. Yeah, good question, very good question. So I'd just like to remind everyone that this research doesn't happen in a vacuum, and we have a great team at the New Zealand Brain Research Institute. And this has been the result of a lot of work over many years from a lot of great people. I'd just like to thank them as well as our funders, which has made a lot of this possible as well. So, I think that's all I wanted to speak about. Well, actually, I mean, I could speak about lots of stuff forever, but try to keep it relatively short and interesting for everyone. I'm very happy to take some questions, but maybe we'll hold questions, and I'll introduce our special guest, and maybe Nicky can tell you about some of her exciting MR-based work, and then very happy to come back and answer some questions. That sounds okay. All right, thank you very much. I'll just get Nicky's presentation up, and I will try to multitask. There we go, great. So it's my pleasure to introduce Dr. Nicky Slater. So she has just recently finished her PhD at the New Zealand Brain Research Institute and University of Canterbury. So she has been, well, she'll tell you a little bit about the work that she's done. She is excellent, dynamic, an incredible addition, and I'm so glad that she has not left the Institute. So she's now working with us, and you're in for a treat because she has some beautiful images and some great work that I hope you enjoy. Thanks.
A little bit lower. Does that work? Yep. Cool. Well, I don't know if I'm the special guest or if you're the special guest. I think you probably are, but thanks for the intro. Kia ora koutou everyone. So I'm just going to talk briefly today about a brain system I've been looking at, and I looked at for my PhD work at the NZBRI called the cholinergic system. But before I started with that, I thought I'd just show a couple of these images which I think are really amazing. These were recently released a couple of months ago by a group at Harvard University, and they took some healthy brain tissue and that they acquired from a 45-year-old woman who was undergoing surgery, and they needed to get to quite a deep area in the brain, and they had to remove a little bit of tissue above that. So they took that healthy tissue, fixed it, and they actually just used 1 cubic millimeter, which is about a millionth of the brain, sliced that into 5,000 pieces, imaged all of those, and then put them back together. So it was the first time this has been done, and they came up with these images, and this is just a tiny part of it. But just to provide some context about what I'm going to talk about after this just shows neurons, or the specialized brain cells that we have that allow our brain to transmit information through the brain, so essentially allow our brain to function. And neurons have a cell body, which are these bulbous sections, which you can see all throughout this image. The colours here are just, they don't look like this in the brain. This is just denoting the size, so I think they've used blue. It's quite subtle, but blue indicates smaller neurons or nerve cells, and the yellows are bigger, slightly bigger ones. So they all have this cell body, and then they have these projections. They often have lots of small projections coming into the neuron, but then very long, well, in the brain, especially in the system, I looked at some very long projections, and there's one main projection from each cell body. So you can see them sort of amongst it all, but this here example is one heading, in this image they're heading upwards. And these projections are called axons, and that's quite important for what I'm going to talk about. So I'll just show you one more of these pictures, because I think they're pretty cool. And actually, I don't know if that's coming through very well, but this is just looking at one of those layers that they imaged. And here they've just coloured the nerve cells different colours to differentiate them. But you can see slightly less obvious, but the cell bodies are these bits here, and the projection axons are going up in this direction. There's a slightly easier look at diagram, just to further explain that we have the axon here, which is going, oh, whoa, that's too soon, going away from the cell body, which is here. And these axons are covered in these sort of globus, about 1 millimetre long, what we call myelin sheaths, and they're made from a fatty tissue. And they actually, when a brain is dissected, if we can do that post-mortem, they appear white because of this fatty tissue, and so we know the areas where there's lots of these axons as white matter in the brain, so it appears white. So we can look at white matter in vivo. Not everyone wants to give us a piece of their brain while they're still alive, so we need a way to look at this in vivo, and we use, as Tracy said, magnetic resonance imaging to do that, to look at structure rather than function with the PET. And this is actually one of our participants in the Parkinson's disease program we have at the NZBRI. It's their brain. So on the left there we have an image sliced this way, and we're looking frontwards. And there's no skull on this, so we can see, and the contrast using this imaging allows us to see the white matter regions as particularly white. So, and then we can have nearer the edges of the brain, grey matter where the information is synthesized and things. So this is pretty cool, and we can actually extract from this just the white matter bits, which will help studying the brain system that I wanted to look at. So on to that, we have the brain cholinergic system. I'll try not to get into too many of the technical details, because it gets technical pretty quickly, but essentially this brain system delivers a chemical messenger called acetylcholine. And we know that acetylcholine is important in regulating sleep, and also especially important in memory and thinking, and attention. So we can, if we want to pay attention to one thing, but there's things distracting us, keeping our attention on that one thing is significantly contributed to by this system. Also, our ability to retain information in the short term, and plan out tasks and then complete them in the steps that we've planned. The system has a big impact on that. And like a few of these brain systems, the actual neuron cell bodies, so those more bulbous bits we saw before, they're actually only located in a few areas of the brain. And for this system, for the cholinergic system, we have one area here in the brainstem, and then a couple of small areas up here in what we call a subcortex, quite deep inside the brain. And so if we see changes, or if there's any damage to these areas, the impact can be really widespread, because the basis of the system is in one really small area. So I've been particularly interested in this, just one part of this system, and it's the projections, or those long axons that go from the nucleus basalis submagnate. You don't need to remember that name. We'll call it the NBM, and I've just circled it in yellow here. And that is a really small region. It's about 1.3 millimeters wide, and the projections from that go really far, like all throughout the cortex. So we know that in Parkinson's disease dementia, from brains that have been studied after people die, that there are fewer cells, these cholinergic cells, in this little region when people have developed dementia with Parkinson's disease. But we don't really know if there's any changes in the system before people have reached that stage. So it's thought that maybe there aren't changes in the system before then, but there could be changes in the axons, so those projections that are going from here all throughout the brain. And the cool thing is we can use MRI to look at this. So using back to that image I showed before of just the white matter parts from the MRI image, we can actually create these amazing images, well, and they'll have practical uses, but they make pretty cool pictures. And we call this a tractogram. So here, where there's bundles of axons all going the same way, some very fancy maths can process this and create what we call a streamline. But we have about 100 million, a billion at least, neurons in the brain, and about as many projection, axons. This system probably has about 1 million or so, but it's many. So we just represent these as a line, a streamline representing quite a few of the axons. And even here here's probably about 1 million of these streamlines, and we obviously can't differentiate any in this image. But we can model them all, so we can use the computer program to show us where all these axon bundles are going, and then we can use that to take out the streamlines or the pathways that we are interested in. Before I go into how I did that, I just thought I'd show this quick series of images that are just showing that we can constrain these tractograms, so all these streamlines, to within the white matter of the brain. So here we're looking at a slice, we're looking from this side at a slice taken through here, so the white ball here is the eyeball. And these images are going to travel towards the midline like this. And we can see that these streamlines and the tractogram are all located within the white matter. So it's pretty cool that we can model this, and these images are massive files, and it takes quite a while for the computer to create them. So this is ended in the midline, and the colours here just denote again where the direction the streamlines are travelling. And right in the middle there we can see that big red strip, and we know there's a pathway there, a white matter pathway that goes from side to side between the hemispheres of the brain, and that's replicated here in this image, which is pretty cool. So what I've done is taken those massive tractogram files and pulled out just those streamlines that are representing those axons that are connected to this tiny region, the nucleus basalis, so between there at least, connecting that tiny region to the whole cortex. So here I've just put a yellow dot where about that tiny region will be. It's much smaller than that, but for purposes of being able to see what I'm talking about. And I've extracted, again, this is in one of our actual participants in our Parkinson's program, and this image is taken looking forwards like this. So in their left, because the images are flipped in MRI, so this is their left nucleus basalis, and we can see here all the streamlines connecting the cortex to this region in this person. So it's slightly less messy. It still looks a bit like spaghetti, but this is actually really useful because we can then look at the various measures we have from the MRI and get numbers from them and compare between different groups. This is another image, slightly more refined from the previous. This is actually from my final analysis of my PhD, where I ended up actually only looking at the connections between the tiny region here, the NDN, and the cortex on the same side. So we call those the ipsilateral connections. And the colours here are actually representing three sort of main directions we think these pathways take in the brain. And that information has come from one study that did some histology. So on a post-mortem brain, they tried to trace these pathways which go everywhere, so it's really difficult to trace. But this has got quite a lot of detail, but I quite like this picture. So this is essentially on the left, this is the histology, this is what they did in the late 90s actually. And I managed to essentially, the detail as I say is quite a lot here, but I managed to replicate that in vivo, showing that this technique can actually try and follow these very diffuse, we call them widespread pathways in the brain. And in terms of what this can tell us in Parkinson's, this is just using the cognitive groups that Tracy described before down the bottom. So we've got the Parkinson's disease participants in our study classified as having normal cognition, those with some memory and thinking problems, and then those who've got a diagnosis of Parkinson's disease dementia. And I did look at quite a few different ways of measuring what we call the integrity or the structure of the white matter going from this tiny region. But in some of these measures, I did find that those people with more memory and thinking problems did have lower, in this case, density of their streamlines, but lower integrity, if you like. So why do this? What can we do with this information? Firstly, we can look at whether the system is actually contributing to the changes in thinking and memory that some people do experience in Parkinson's disease. And the original idea with this work was that perhaps this could be an indicator if we see early changes of future memory and thinking problems and Parkinson's. And that's really still to be determined as this particular cohort, the research is ongoing in terms of that outcome or longer term memory and thinking assessments. And also in terms of how it can be applied clinically, if the system we know is involved in those memory and thinking problems that some people experience, there may be medications that can help, and if we can prove that there are changes in the system, that can be used in a clinical situation. So, yeah, that's all I have to add to that. I don't think I have any other slide. I do need the same photo again. But yeah, we're happy to take questions. Thanks for listening.
I have a microphone that I can rove around to help us with questions and answers.
He's consulting his book.
Must be a good question in there, is there?
I'll take another question.
Any other questions while Mrs. Bonnie is?
A few years back, I've been diagnosed with Parkinson's, tell, but a few years back, someone in the Brain Research Institute, whatever, said that there was a 30% chance of people with Parkinson's developing dementia. I just wonder whether those statistics are true today.
That is a very good question. So that everyone heard that, yeah? Yeah. So that has been tackled by our statistician Daniel Mile, Dr. Daniel Mile. He has been looking specifically at that because he wanted an answer to that particular question. When we look at the data that we have available to us across the nation, so this is big data from some of the databases available, we see that that's probably about the number that we come up with as well, about that. Again, there's a little bit of variability either way, but we think that that's probably about the number we come up with as well. Yeah.
Thank you.
So I'll just answer this. How long does it take dementia to develop? A very good question, a very challenging question, and a very frustrating question. A, doesn't happen to everyone. B, it's incredibly variable. And you can see some people who are diagnosed and then progress to dementia very quickly, on the order of a few years. And you can have individuals who maybe never develop dementia. So we're going decades after a diagnosis of Parkinson's and they're going fine. So it's incredibly variable. And a lot of our work is trying to find out why we see that variability, and if then we're able to address why some people do and some people don't. So it's a very good question. It's a very challenging and frustrating question because it's highly variable. Very short for some people, very long for others, and not at all for others. Question?
Obviously, we as a Parkinson patients, we are looking for hope. We don't have much time, and time is very precious for us. Any hope we can get, we grab with full hands. Now, I have a book here which indicates the impact of exercise on the progression or prevention of Parkinson. Especially, there's a timeline here. In the last 2 decades, I would say, there have been quite a few scientific researches which, without any doubt, correlated the slowing of Parkinson's symptoms, cognitive and non-cognitive, motor and non-motor symptoms, with rigorous exercise. The first one, which came in between 2012 and 2015, has been run by Professor Daniel Korkos in the United States. And he ran an exercise experiment called Spark X2, which clearly indicated high impact exercise. By high impact exercise is you increase your heartbeat over 80% of your maximum rate for an extensive period of time and continuously. During his experiments, he used treadmill for 2 to 3 days a week for a period of each section, 30 minutes, and over a period of 6 months. There was a clear indication that the people who have been submitted themselves to rigorous exercise have got significant improvement in their symptoms. This exercise has been now repeated. It was so significant, which is, as we speak, running as a Spark X3 between 2023 and 2028, will further improve the hypothesis. This is what happens in the United States. In Holland, and this is spearheaded by Professor Bastiaan Bloem, they did a study
Who
called...
will be here. Bastiaan Bloem will be here.
Oh yes, he will be here in August. Yeah, he will be here. He will be in August. Between 2015 and 2019, it's called Park and Shake. He used stationary biking for the same type of exercise, but he used also the imaging techniques, I think mainly MRI. And this is now being repeated between 2016 and 2018, phase 2. He significantly indicated, by using MRI techniques, the shrinking of the raw proteins, mainly alpha-synuclein. So, do you think that this is a real chance for us to get on our butts and improve our heartbeat, for as much as we can sustain, and can we get some improvement? Or is your research, with your now very sophisticated imaging techniques, will do something in this direction here in New Zealand?
Yeah. Do you want to have a go? Well, yeah.
Don't you come and say that?
Oh, if you want to, yeah.
Funding in New Zealand is few and far between, so I think if someone was funded, to do it, then that would be definitely an interesting question. But that's what immediately comes to mind is we're so limited in how much we can actually do here.
But this is very crucial for us.
A lot of it's crucial though,
And the outcome of it was, any exercise base should be built on the cardiovascular first. The other ones, coordination, balance, flexibility, should be complementary.
Yeah, I mean, there has been one study at Interberai, Megan's work on enrichment with exercise, right? Yeah, I don't think it's been published though.
We haven't published that one,
No.
but I mean, just as a general response, I would agree. I mean, I think that there has been a raft of information about exercise and the benefits of exercise to people with Parkinson's beyond people with Parkinson's. So in older age, in a number of different conditions. I think there's been a blanket positive across the board number of different exercise programs which have shown benefit. Specifically to Parkinson's, I actually don't know the Spark trial, but and it sounds familiar with a number of the other ones that I've seen, where there's been a bunch of the cycling stuff which you mentioned, and aerobic, both aerobic and strength training. So anaerobic and aerobic exercise definitely has benefits, which I have also seen and I agree with. And so Professor Tim Anderson, who I'm sure most of you know who he is, I mean, he always comes back to the same things of saying, physically active, mentally active, socially active, and eating a healthful diet. And I think across those 4 things, we do have quite good evidence to suggest that those are helpful, not just for everyone, but can specifically be helpful for Parkinson's disease as well. So I would agree with you, absolutely. Those trials do suggest that exercise is a benefit for people with Parkinson's disease. Whether the high intensity or like the HIIT workouts, the high intensity interval training type of workouts where you get the really high increases within a healthful scenario do look to be beneficial. But that's not to say things like, there's been a bunch of dancing work showing that dancing is quite effective and helpful. I think that there's evidence to suggest both those high intensity workouts, aerobic workouts, some anaerobic strength training is helpful. Again, you don't have to be Arnold Schwarzenegger, 70,000 kilos. Small anaerobic weights can be quite beneficial as well. So I'd agree, yes, that is some quite nice information and evidence there. And whether we can implement some of those things in New Zealand, we definitely would like to. We, as Nicky alluded to, we did have an enrichment program, which was looking at some occupational specific physiotherapy, as well as some cognitive therapy, as well as some social engagement. So we are certainly aware of that, and we'd like to do more of that, and I think it's a good question.
But this school of thought researchers, they specifically emphasize and focus on the high impact. I mean, high impact is the key.
Yeah.
Is the fastest, more effective way of reducing the symptoms, of delaying the progression. And they have a theory behind that they generate neurochemicals in the cortex which reduce the agglomeration of all these raw proteins what you are talking about.
Yeah,
And the newest research, by the imaging techniques prove that. I'm not a scientist, I'm just reading, because I read everything, which can give me hope, to improve my condition or spread the word.
Yeah.
So this is not, in general, do a little bit walking. It's not. It's intensive. I mean, we cannot walk fast enough, so walking is not for us. But we can do stationary biking.
Yeah.
It's not affecting our balance. We can hike the pedals like hell, and let the heartbeat, pump like hell, and this works according to these people.
Yeah.
And
Well,
if this is the case, this message should be spread out throughout the whole community, because we don't have time to lose.
Yeah. No, I think it's a good point. I will definitely read those Spark trials. I haven't read those, so yeah, I look forward to it.
Thank you. We've got another question. Question.
Over here. Is there any research similar to this for MS?
Is there any research similar to this but for MS?
Yeah.
So the work that, the greater we have done at NZBRI on the MS side of things most recently has been around epidemiology, so has been around incidence and prevalence and trying to get accurate numbers of people affected by MS across North and South Island. So that we have a history of that. There was a decade ago, and now recently followed up on that. But in terms of this type of work, so imaging work, we don't currently have any MS projects with imaging, or actively having participants come in to participate. So we don't currently have that, which is, I think, is a shame, I think, because we certainly did have a quite active MS group a few years ago. So unfortunately, no.
Yes, can I just ask you, I was diagnosed about 5 years ago with Parkinson's, but since then I've developed neuropathy in some of my fingers and my toes. Is that part of Parkinson's, or is it different?
Now that's certainly a clinical question, which I'm a scientist. Fortunately or unfortunately, depending on how you view that. My understanding is that is something that happens in Parkinson's, although again, I can't advise anything there. But, yeah.
Are there different sorts of Parkinson's you can get?
Are there different sorts of Parkinson's that you can get? Do you want to ask that?
You can answer.
Okay. That's a very good question. So I think there's an easy answer and then there's a more difficult answer. So I think the easy answer is we have Parkinsonian disorders where there's some sort of movement impairment, right? And we have what we call idiopathic Parkinson's, which is the majority, but then you'll have atypical Parkinsonian disorders like multiple system atrophy or supranuclear palsy, and there's a number of different those. So that's the easy answer, in that those are well-defined, characterized. They can be difficult to diagnose, but agreed upon in the clinical world that these things exist and that they're different from each other. The more difficult question is within, let's say, Parkinson's disease, what we think of as Parkinson's disease, there is an incredible amount of variability. And there has been a lot of work trying to characterize that variability, trying to subtype individuals or subgroup individuals to say, ooh, you look more like this symptomology, or you look more like this symptomology. And does that mean that pathology is different or that disease progression is going to be different? And that is really murky because it's difficult. And people are spending a lot of time trying to sort through all of that. So it's a really pertinent question. And unfortunately, yep, I think it's probably a perfect reflection of, unfortunately, our understanding of the situation at the moment. Yeah, it's complicated.
I had a scan about 3.5 months ago, and they called it a DET scan.
Yep.
What's the difference between a DET and a PET? Is it the same?
That's a wonderful question. What's the question? What's the difference between a DAT scan and a PET scan? So, historically, a DAT scan has been to look at dopamine function, which we know is generally affected in Parkinson's disease. So, a DAT scan generally uses a technique called SPECT, single photon emission computed tomography. So SPECT, which uses radioactive material, but it's a different radioactive material than PET scans. So it's a different type of imaging. It's called SPECT imaging, and it allows you to look at dopamine function. However, so that's available at Christchurch Hospital. And so we do DAT scans every day to look at dopamine function. However, now there are PET equivalents for DAT, so to look at dopamine transporter or dopamine function. And those are currently attempting to be funneled into the New Zealand system. So at the moment, it's SPECT. It's a different technique to PET, which allows you to look at dopamine, and potentially in the future, we'll be looking at dopamine with PET as well. If that helps.
Sounds like your scanner's at Southern Cross. Is that right?
The PET scanner is at
Yeah,
Southern
so
Cross.
that's privately funded. I'm just wondering how people get to be referred, or who refers into you, who's funding it, how much does one of these cost to do?
That's a good question. So now the New Zealand health system will fund lots and lots of metabolic PET, and so that's mostly in the cancer area, right? It's an incredibly useful tool for cancer. So that's where it spends 90% of its time. All of these are still research techniques. So the amyloid isn't funded, tau is definitely not funded, and some more of these, the novel PET, are not funded and are only research at the moment. I think there's a general push to get something like amyloid funded, especially from an Alzheimer perspective, especially from an early onset or young onset cognitive problems. That is when it can really be helpful to sort out, is this Alzheimer's, is this not Alzheimer's, and if it's not Alzheimer's, what do we do about it? If it is Alzheimer's, what do we do about it? That's not currently funded, but we are hoping that there will be a pathway for that in the relatively near future.
Thank you. Thank you, Tracy. Thank you, Nicky. I think it's time to wind up. That was, thank you so much for all your awesome questions. They were really good questions.
Yes, they were.
Thank you both for just for your generosity of spirit about what you bring to your mahi, your work, because I can really feel how this area takes quite a lot of passion and interest because it's quite complex for my brain to understand. But you've really shown us a little bit about how that different imaging techniques might be able to, can be applied in Parkinson's research and is being applied. And I guess it just seems like there's still a lot of hopefulness for what we might learn into the future. And yeah, we really appreciate you giving up your time and coming in your day to teach us a little bit about what you do and to demystify, I think, especially the differences between the different types of scans, because we do get a lot of people have had things and you don't know why. Why did I have that and not the other one? And should I have that other one? And how do I go about getting that? So I think that's really helpful for us to just grow our understanding and see where research might take us to understanding the impacts of dementia and Parkinson's as well as Alzheimer's and other conditions. So thank you very, very much. I just, I've got a little gift here for you, Tracy, thank you. And Nicky. I think you too. So we'll just give them a clap to thank them.